Hello, everybody. As Michelle said, I am Livia Civitelli, and I'm based here at the University of Oxford. And I'd like to bring you toward this journey about the connection between Parkinson's disease and the gut, and what stool samples can tell us in research. So in 2012, my husband's father was diagnosed with Parkinson's disease. And before the diagnosis, we were used to notice that he had the REM sleep behavior disorder. So this disorder was affecting him in the sense that he was not laying still while sleeping, but he was actually acting out during his dreams, for example, through vocalizing or very complex movements. REM sleep behavior disorders, in shortened RBD, is a parasomnia. That means that it is characterized by events that disrupt the sleep. And these events are mainly driven by the fact that there is an absence in muscle atonia. But Parkinson's disease does not just appear as it was for my husband's father. In the case of RBD, there are subtle changes that start even before you receive a diagnosis of Parkinson's disease. And these subtle changes affect the body before you can really reveal these in terms of symptoms that actually affect the brain. And this period can last for a very long time. It can last up to 20 years, and it's called a prodromal period. And in this period, as I say, these subtle changes are there. They start to appear, but you don't really notice any very evident effect in terms of being diagnosed for Parkinson's disease. And these changes can be followed by either molecular imaging or by collecting cerebral spinal fluid, in short CSF, which is the clear liquid that surrounds both the brain and the spinal cord. And usually this is collected for diagnostic purposes. But CSF is not only a window to the brain, it can also be tested in what is called the seed amplification assay, which is, in shorten, is called SAA. And this is a very powerful technique in the sense that it's made to test the presence of very small, tiny amounts of proteins that are related to Parkinson's disease that have taken up a wrong shape. And these, as you can see at the beginning, these are mainly invisible, but as soon as you take on this kind of technique, you will end up in an endpoint where you can see this tiny, small, misfolded, as we say in a technical language, proteins that become visible, therefore can be detected. Many different biosamples are tested in SAA. However, CSF is the most reliable, and it has been went through a long, robust validation, and also because it gives very robust results. Parkinson's disease does not appear the same for everybody. So there are people who experience symptoms that are more related to the brain, and in this case we talk of what is called the brain first hypothesis. But there are also people who experience symptoms that are more related to the periphery, meaning that they affect the brain only in a second wave, I can say. And this has led to the hypothesis of the body first, where what is affected firstly is the gut, and therefore we call it as periphery, and then it comes only in a second part affecting to the body. To the brain, sorry. So if we think that the gut can hide some of these subtle changes, our aim is to study the fecal samples. And this is because we think that we can capture this early sign in a simple, non-invasive way. the main protein that is related to Parkinson's disease is alpha-synuclein. Alpha-synuclein is a protein that whose function is not really known yet, but it seems that it can help brain cell-to-cell communication. And when it takes on the wrong shape, it starts aggregating and it clumps together. And these aggregates will affect the normal function of the brain and also of the cells. So here you can see, like in this image, that different example of these aggregates that are highlighted with the yellow arrow. And in our studies, we have found that in PD samples, in particular in stool samples of 45 different PD versus healthy control, we found many more presence of this aggregated form of alpha-synuclein with respect of healthy control. And it's I think you can see only by eye that we have many more bands in this in the left part, which is the PD versus the healthy controls. And these bands, these aggregated forms, were revealed by antibodies that are specific for capturing this misfolded presence of alpha-synuclein. So, as you can imagine, stool samples are very complex. It's a very complex matrix, and obviously proteins are not there alone. There are other co-founding materials, so it can be very difficult to interpret this result. Therefore, we have decided to focus on extracellular vesicles that are isolated from the stools. Extracellular vesicles are tiny fluid-filled sacs that are released by all human cells. They have the property that can circulate, so they can also reach very distant area from where they are released, and they can have the peculiarity that since are released, as you can see here, they are released from mother cells, they have the property to really mirror the mother cells itself, meaning that they can carry information that are really and easily transposable to the mother cells. What we have done is to apply the seed amplification assay to both stools and extracellular vesicles. So in here you can see that we have applied the seed amplification assay to stools, and as before we had 45 PD samples and 35 controls. The sensitivity and specificity was not very high, meaning that we didn't get many PDs to get a positive signal, and we also have healthy controls giving a signal. This can be like a sort of information about false positive. But here, when we have used extracellular vesicles, we have found that 5 out of 5 of these EVs extracted from the poo of PD samples give amplification, which gives a sensitivity of 100%. However, specificity was still not very good, meaning that 2 out of 5 amplified. Obviously, the numbers is very low, so the aim of the future work will be to increase this number to get more statistically power. And another thing that we have done, it was to analyze the content of the EVs in terms of protein content. And what we have found that is very interesting is that here on the right panel you see the PDs, and here you see the healthy controls. What you can simply see is that here you have many more green proteins than here. These proteins are related to the immune system, and you can see by eye that in the case of PD we have proteins that are linked to the immune system that are lowering PD, meaning that the immune system in some way is struggling in PDs. This is something that goes a little bit against what is known in the literature, that PD is an immune-activated disease, but you need to consider that here we are working with fecal samples, we are working with the periphery, so it can mean that we get different results from the brain versus the periphery. So to conclude, I can say that Parkinson is a whole body disorder. It's not only linked to brain, but we have started getting information about the fact that there is a condition affecting the whole body. And also we are very like happy to be focusing on gut and fecal samples and extracellular vesicle, also as a non-intrusive human samples that can be used instead of CSF, as we know that collecting CSF can be very intrusive for patients. And we also hope to uncover these early signs that can lead to a more personalized care. And thank you.